Hello everyone, I am Priyanshi Chaudhary, a volunteer at Shivi Forensics, and today I will be discussing about the serological examination of blood and blood stains. But first, we need to understand what is forensic serology. It is a subfield of forensic biology that deals with the examination and identification of biological evidences. It focuses particularly on the biological evidences such as blood, semen, saliva, or urine. So in this field what happen is it helps us to determine the presence of the biological fluid, its identification, origin and the analysis of blood stain patterns. For example, when you go on a crime scene and you see any fluid present on floor or a clothes, then you will look at the physical state of that fluid and you will determine whether it is biological fluid or not based on that. Afterwards, presumptive and confirmatory tests would be performed on it to determine whether this fluid is actually biological fluid or not. After the test come positive, then species identification is done on that biological fluid to determine the origin of the fluid, like if it is blood, to determine whether it's belong to human or it belongs to animals. This field does not deal with the examination of DNA. Now we will discuss about the collection procedures that are used to collect these biological evidences from the crime scene. So when we talk about blood, semen or saliva, usually it is present in two states on the crime scene, either in liquid or either it's going to be in dry state. So if we have blood and it is found in very large quantity like in pool, so what we will do, we will take a syringe and we will collect the blood in EDTA vial. Now it is important to collect it in EDTA vial because EDTA is a preservative. But if we have these fluids in very small quantity, then what we will do, we will take a sterile swab, cotton swab, and we will put it on the fluid and we will let it absorb all the fluid in it, and then we will air dry it and pack it in the paper bag and send it to the laboratory. Now, if these evidences are present in dry state, then the first method that could be done is scraping. If possible, depending on the surface they are present on, we will scrape the biological fluid from it. If that is not possible, then we can use the second method, which is double swab method. Now in this method, what we will do, we will take a wet swab and we will rub it around the surface on which our stain is present. It will moisturize the cells of the stain or the fluid. Afterwards, we will take a dry swab and we will again rub it on the surface. So it will collect all the cells. Now these both swabs are packed and sent to the laboratory separately. Next, we can also do one thing, we can just cut out the part on which the stain is present. For example, in case of semen, if it is present on clothes, then we will just cut out that cloth area on which our stain is present, and obviously because we are cutting out a cloth area, we have to take a reference area of cloth also, and we will pack both of them separately and send them to the laboratory. Now after these evidence reach the laboratory, different tests are performed on them. The first one is presumptive test. So it is the initial screening test used to detect the possible presence of a specific biological fluid. So what happens is when any of the fluid come to the laboratory, you have to perform the test to determine whether it is biological fluid or not. So afterwards, when you determine yes, it is biological fluid, then you will perform a confirmatory test which is more rapid and which has high certainty and which is highly specific, and this will help you determine that if it is biological evidence, then which type of biological evidence it is. Because in presumptive test, there could be false positive results also. Now, if we looked at the whole flow work of the examination of biological fluids, let's take the example of blood only. So here you can say that we have a blood stain and then we will perform visual examination of it either on crime scene. Mostly it is done on crime scene. Then afterwards we will perform presumptive test to determine whether it is biological fluid, if it is whether it is blood or not. After we are done with that, we will perform confirmatory assay. which is to determine actually it is blood or not. मतलब this is give us more surety. Confirmatory test is more particular and this give more surety. Along with it, we will perform species identification. So this test is done to determine the origin of the blood, whether it belongs to human or animal. So once we determine yes it belongs to human, then we will perform DNA profiling to determine that which human it belongs to. Now because of DNA profiling, we will get individualization because now it will be specific to a particular individual. So if we talk about the serological examination of blood and blood stains, as you know, the first step is visual or physical examination. In this, if we can see the blood or stain with our naked eyes, then we will just see its color, whether it's brown or reddish brown or not. Afterwards, we will check if it is in dry state that what is the physical texture of it, whether it's powder, whether it's crystal or not. But when we are talking about blood, it's usually get washed off in crimes, so we can't see it with naked eyes. So in those cases, we will use UV light. We will use the UV light of frequency 230 to 269 nanometer and see on which part the blood was present or not. Next, we will perform presumptive test. So the most commonly used presumptive test is tetramethylbenzidine. In this test, we will take reagents which are as to prepare acetic buffer, we will take sodium acetate anhydrous, get glacial acetic acid, distilled water. To prepare working solution, we will take TMB and acetic buffer. To prepare working solution 2, we will take 3% hydrogen peroxide. Now the main component of this test is TMB. Why? Because as you can see in this picture, we have tetramethylbenzidine, which is colorless. But in the presence of hydrogen peroxide, it will lose two of its hydrogen and make a double bond. And then it will become tetramethylbenzidine diamine. And it will show blue-green color. So in this test, what happens is you take a swab and then you put it on a filter paper or a spot test paper and add few drops of TMB solution or working solution 1. Then after it, you will add few drops of hydrogen peroxide on it. So after adding hydrogen peroxide, you will see that a blue-green color is coming. If it is actually blood, then a blue-green color will come and it will show the activity of peroxidase. Now peroxidase activity indirectly indicate to the presence of hemoglobin. But it is not directly indicated. It is only indirectly indicating. So that's why this is presumptive test. Next we have phenolphthalein test. This is also called Kessler-Mayer test. In this we have three working solutions. First is ethanol, second is phenolphthalein, distilled water and ethanol. Third one is hydrogen peroxide. Now the main component of this test is phenolphthalein. Because when you put a swab on the filter paper and then you will add few drops of ethanol on it. Afterwards you will add few drops of phenolphthalein solution. So when you add few drops of phenolphthalein solution, you will see whether there is any color change or not. And if there is no color change, then you will add hydrogen peroxide. After adding hydrogen peroxide, you will see there is a bright pink color which will come. Now this bright pink color will indicate again the activity of peroxidase and it will indicate the presence of hemoglobin. The next step is confirmatory test. So in confirmatory test, the one we use is Takayama test. In this, we have reagents such as standard glucose solution, sodium hydroxide, pyridine and distilled water. See in this image, we have hemoglobin and in the presence of glucose, sodium hydroxide and pyridine, what is happening is the Fe of the hemoglobin is getting attached to both the pyridine and it is becoming pyridine hemochromogen. Now because of this, we can see pink needle shaped crystals in the microscope because this form a complex together. Now this pyridine is directly attaching to the hemoglobin, so it clearly shows that yes, in this fluid a hemoglobin is present, and this become confirmatory test for blood. So the whole procedure of performing this test is as you will take the swab or the stain on the microscopic slide. You will put few drops of digamma reagent on it, and then you will cover it with a cover slip. Afterwards, you will heat it at 65 degrees Celsius for 10 to 20 seconds, and then you will allow it to cool down, and then you will observe it in your microscope. So as you can see here, pink color crystals, you will see these crystals. Next we have Teichmann's test. Now this test is same as the previous one. The only difference is in this test the hemoglobin is attaching to the halide group of atom in the presence of the salt of that halide group and the glacial acetic acid. So here in this case you can see that hemoglobin is attaching to Cl, chlorine, in the presence of sodium chlorine and glacial acetic acid. So the procedure is same. We will take our stain or the swab on the microscopic glass slide. Then we will put few drops of the reagent, and then we will gently heat it at 65 degrees Celsius for 10 to 20 seconds. We will allow it to cool down, then watch it under microscope. Now in the previous one we were seeing pink color needle shaped crystals. In this we will see brown color shaped crystals, and these crystals will show the presence of hemoglobin. The next step is that we perform is spectrophotometer estimation. So in this we will use UV spectrophotometer, and we will take our sample in working solution 1 and incubate it at 37 degree for 15 to 20 minutes. Afterwards we will add the working solution 2 in it and change it to the cuvette. Now we will add the solution to the cuvette of UV spectrophotometer. Now you will run the UV spectrophotometer at 560 nanometer or between the range of 500 to 600 nanometer for 5 to 10 minutes. After you will be done with this, you will see there will be two peaks at 558 nanometer and 529 nanometer. Now these two peaks will determine that either this fluid have hemoglobin or this have derivatives of hemoglobin. So all these tests has directly given us the identification of hemoglobin. That's why these are confirmatory tests. Now we will perform the test for determining the origin of the biological fluid. So in this, for blood, we will perform ABA card. Now ABA card is a immunochromatographic assay. So why it is immuno and chromatographic? We are use chromatographic technique principle in it, and with the help of antigen antibody reaction. So that's why this is immunochromatographic assay. Now this is used to detect human hemoglobin specifically, and it utilizes monoclonal and polyclonal antibodies. So what happens is we have WBC and they produce B cells. Now these B cells produce antibodies. So when these B cells produce antibodies, they produce two types of monoclonal and polyclonal. Now monoclonal are the ones who will attach to a single site or a single specific antigen. They will not attach to multiple. They will attach to a specific region or a specific antigen. But on the other hand, polyclonal will attach to multiple regions. So that is the difference between the both. And the basic principle of it is that the whole technique is based on antibody interaction. It will form like a sandwich of antibody, antigen, and then antibody, and based on it we will get the color, and on based of that we will determine whether our biological fluid, which is blood, belongs to human or not. So we have, as you can see here in this picture, we have three zones. One is C, another one is T, and the last one is S. So in S zone, what we will do, we will have monoclonal antibodies, and we will have those in there, and then we will add our hemoglobin antigen. Basically we will add our blood has hemoglobin, it has antigens in it, so we will add it. Now because it is a chromatographic technique and we have this stationary phase also. So T zone is a stationary phase in which we have polyclonal antibodies. So obviously our mobile phase is moving upwards. So in S zone, this antibody of monoclonal and the human hemoglobin antigen has already made a connection. They have already interacted, and now in T zone, another antibody which is polygonal will get attached to it, these twos and form a sandwich like structure. Now, these antibodies, these are labeled because we are producing them in the laboratory. So these are labeled. So when they will go to the C zone, they will either produce pink light or not. Now, based on this, whether they are producing pink line or not, we conclude our result. So if you have two pink line, C and T both zone, then it is considered that we have positive results. Blood belongs to human. If we have line only in C, then it is not the blood which is belonging to human. If we have no line or we have line in T only, then again our result is invalid. Now we will talk about the forensic significance of the serological examination of biological fluid blood. So first, what it help us identify? It help us identify whether we have blood or not. Like when the fluid that we have found on the crime scene is blood or not. Then it help us determine whether it's human or animal blood. It also help us locate blood at the crime scene. When you see, when you are not looking, when you can't see blood from your naked eyes, then you will use UV light and see the blood. So it also help us locate that. Next, we will reconstruct crime scene based on the analysis that of blood patterns that is done by forensic serologists. And we also support legal evidences with the help of this. In the court, when we will show the legal evidences that the fluid that was present was actually blood, it belonged to human, and that this is the DNA profile, then it will support legally. So this is the whole sum up of the serological examination of blood and blood stains. Thank you.